Now I'll make a mini lecture for each of these topics. So each uses a figure from chapter 20, and you can take a look at the figure, and the figures are well explained, but then there's also plus the textbook section that you can read that describes these. And so the first method that we're going to learn about is called DNA sequencing, and that's how the method that's used for scientists to actually read the letters in the DNA code. And that was discovered first done about in 1975, and since then there have been improvements in the rate that the DNA sequence can be read, and now there are billions and billions of letters that are available in repositories on the web for scientists to compare. And this takes us back to the very first images that we had in our class and the very first video that was about insulin and how humans synthesize insulin from amino acids that we get in our diet. And so this is just this story that was talked about in that very first video of the course, where the insulin gene sequences can be compared between two people, like Nancy and Bill. And then it zoomed in on this idea that that chain of amino acids was based on the coding region that we learned a lot about in our course since the first day, and then that coding region is represented in the DNA. And so now scientists can read the DNA code and they can get the sequence, and that sequence can be used by a computer to predict the primary amino acid sequence of a protein. And that DNA sequence has been read for humans, and that would be the human genome, and it's been read for many humans. Hundreds and thousands of humans have had their genome sequenced, and that's significant because we can draw comparisons and look at different alleles represented in the different populations and regions of the world. And that's being done a lot to understand humans, and also recent human evolution can be studied by studying humans and comparing to, say, our primate relatives like that. So that work is done, and we learned a little bit about this idea that work is done on the website for the National Center for Biotechnology Information, and that comparison, that kind of word search comparison, is called blast search. And so DNA sequence leads to huge amounts of data that can be sifted through and studied and analyzed by using things like blast searches. This is just the output from a blast search, again from our very first class, talking about finding sequences like the sequence for the human insulin gene amidst all of that information, in the same way that you do a word search, or the same way you find websites by doing Google searches. It's the same logic, and we can have a huge amount of data stored, and then we can search through it to find the things we're most interested in. So here we are now comparing humans and searching the chicken genome to search for insulin in the chicken genome. And you can do that by studying things that you already know a lot about, or you can study a new protein, and maybe you would first study it in fruit flies, and then look for, by blast search, look for an ortholog, and we learned that term during this course. Search for an ortholog in humans, like that. And so where did that information come from? And it came from an instrument called a DNA sequencer, and a sequencer is an instrument that's used to read that code. And so this figure in chapter 20 takes advantage of this DNA replication that we just learned about in chapter 16. And a reaction is set up in the instrument in test tubes to allow DNA synthesis to occur in the test tube. And as the DNA is being synthesized, the strands are getting tagged by fluorescent tags that will allow us to read the sequence, because each of the 4 letters has a unique colored tag with it. And so having a spectrophotometer read the colors as they're being added to the DNA will allow us to read the underlying sequence. And so the power of DNA sequencing comes from what we just learned about in chapter 16, replicating DNA and doing that in a test tube. That's being done. Here's one DNA sequencer. Inside that sequencer, there are hundreds of little tiny test tubes, and each test tube holds a DNA sequencing reaction. So it holds DNA polymerase and all the elements that we learned that DNA polymerase needs to get the job done. This leads to just a huge amount of information really fast, and this is this reductionistic approach that this semester has been based on that is really driving much of biological discovery. And that in what happens in the test tube is depicted in this figure. So the book talks about this figure in the text and then shows this figure and describing it. And so here's the setup. Here's what goes in the test tube, and what goes in the test tube is, the bottom line is, its DNA polymerase and the things it needs. And we just learned in chapter 16 about what it needs to do its job. It needs to add, you need to add a template, you need to add a primer, and you need to add these nucleotides like that in order for it to do its copying. And it's important to realize that we don't just add one DNA polymerase molecule and one of each of these, but there are millions. Even though it's a really small reaction, we've learned that the numbers in biology are big, and there would be millions of these reactions happening in each test tube. And that helps you understand what happens, because the product gets tagged, and it's showing multiple products because there are millions of these made in each test tube. And here's what's going on. In addition to the normal nucleotides needed by DNA polymerase to get its job done, these all are also added, and these are like broken or defective nucleotides. And they each have the corresponding unique glowing color associated with them, so that extra tag of a fluorescent label. And then they are said to be, another name for these is they're said to be chain terminating, and so that's how they're broken or defective. If one of these gets added to the chain stops growing. And so that's depicted here. After the incubation is done in the DNA sequencing instrument, the products in the test tube look like this, where there's the template and DNA polymerase starts to copy it, but as soon as it grabs one of the broken dideoxynucleotides, it stops. And so this DNA polymerase was processive. It grabbed hold and it added a T, a T, a G, and a T, and those were all the good nucleotide precursors, but then it accidentally happened to grab a dideoxy CTP chain terminating nucleotide, and that one stopped there. And this is just showing that that happens at every nucleotide, because there are millions of different DNA polymerases in the tube. There are chains that got terminated at each of the letters. And so this is what the product looks like, is we have sort of a ladder of labeled strands, and we have strands that are this length, and all the strands that are this length end in a C, and that strand will glow with the associated C color. And so to read the sequence, to do the analysis, this bottom part of the figure, what scientists do is they look at the length of the nucleotides. They do that by taking the product and running it through a gel under an electric current. And in a gel and an electric current, the shortest fragment will move the fastest. And so at the bottom of this gel, there's just a spectrophotometer on a little electric eye watching what color comes out of the tube. And the first color that comes out is pink, and then green, and then purple, purple, green, yellow, pink, like that. And they can know the original chain, the original dideoxynucleotide colors, and they can interpret the colors to be the corresponding nucleotide letter. And so that's how the actual reading gets done, is the spectrophotometer eye watches the color, and the color is associated with one of the original nucleotides, and so we can read the chain. And that's getting done in these instruments over and over. And it gets done really fast. And in the process, DNA polymerase can read about 700 nucleotides. And then it just is impossible for it to go much further in this, to be read much further in this situation like that. And so the last thing to mention about this figure is that scientists have to set up several of these instruments, because each one is collecting a relatively short piece of data that then is used by a supercomputer to find overlaps between what this sequencer read and what another sequencer read, and extend those longer and longer, ultimately getting them up to 100 million nucleotide letters long. So it's an amazing job for supercomputing to line up all of these products. And you're not assigned to look at this figure, but you can look at this figure just if you're interested to learn a little bit more about the sequencing process. It's a big computational effort, and that's just shown here. And the process that scientists do is they refer to it as shotgun sequencing, where they randomly grab bits of DNA and sequence them in those instruments, and then a supercomputer assembles all the overlapping fragments into long, contiguous sequences. And that's what we want to get out of that first method, DNA sequencing.